So this is the experiment 2, in which we'll be observing ECG waveforms of subject, that is human body, using lead 1 of a standard bipolar leads configuration in real time mode. So this is the ECG cum heart rate monitor. This in fact is a trainer which will be used in order to acquire the signals from human body and will be plotted on a DSO. So if we look into this trainer or monitor, so on the figure that is shown on the trainer, we have Ithoven's triangle drawn and the three types of lead connections which are employed. So experiment number 1 deals with lead 1 only, and that is why we will be connecting the electrodes according to the lead 1 arrangement. So in case of the lead 1 arrangement as shown in this figure, so according to Ithoven's triangle, we will be connecting the negative terminal on the right arm, which corresponds to the red electrode, while the blue electrode corresponding to positive will be connected on the left arm, and then the black electrode that corresponds to the common or ground will be connected to the left leg. If in case we needed to perform lead 2 connections, then in that case negative, that is red lead, will be connected on the right arm. The positive lead corresponding to blue will be connected to the left leg, and the ground will be the left arm. Likewise for lead 3, we have positive here, negative here, and the grounded will be right arm. And there are figures drawn for different lead connections. For lead 1, we normally will be observing this type of waveforms. For lead 2, this type of waveforms are observed, while for lead 3 we have this type of waveform. And this is the panel in which the controls and blocks are represented. So we will normally be obtaining or acquiring the signals from the electrodes from the human body through electrodes, and those electrodes will be connected here. So we have input from electrodes which will be pre-amplified first, then there will be filtering done in order to filter out the unnecessary or unwanted signals or the noises. After that, then there comes into picture the amplifier, which will be amplifying the filtered signals. Then there are more blocks which are in fact these blocks will be used in another experiment, but in this particular experiment we will be acquiring signal from the output of the amplifier. So we will be connecting the lead of the DSO at the output of this particular amplifier in order to look how the waveforms looks like. So this is the DSO which will be used in this experiment, and in order to understand how this functions briefly, this had been discussed in the previous experiment. So in order to know how this particular DSO works, it is suggested that you go through the video of the previous experiment. This is the experimental setup that was done while the experiments were performed. So this is the trainer kit, the ECG setup. Then we have DSO here, and then there are electrodes in yellow, red, and black. Sorry, we had the positive electrode in the form of yellow, not blue. So this is a correction here. We have 3 electrodes, yellow, red, and blue. Black, where black is the ground, the red one corresponds to negative, and yellow corresponds to positive. So here is again the experimental setup, in which we have shown the electrodes connected to a subject, and the signals will be acquired from the ECG signals will be acquired from this subject and will be sent to this trainer kit, and from this kit or monitor we will be sending the signal to the DSO. So let's see how the signals are plotted on the DSO in the next slide. So these are the ECG signals from the subject. So in this picture now you can see that there were artifacts. So let me show the artifacts. This particular place, this particular signal shows the movement artifact when the subject was moving. and when the subject is still, no movement is observed, then you can see the pure ECG signals which were recorded. Now you'll have to submit the report addressing the following questions. The first is, what are the different wave amplitudes PQ, RST in lead 1, and second is, why lead 1 is called as bipolar.